master of none. So I'm really glad you guys, the organizers, asked me to come speak. I don't really do anything with security per se. So I titled this process, and I put security in because I said I should make some point of why I'm speaking here. So I kind of end with some thoughts on how this might apply to security, but I'll primarily talk about the process by which my lab has started to propose how we can make low-cost, continuous flow CNC milled microfluidics in service of synthetic biology applications. Just a little bit about myself. My primary appointment is in electrical and computer engineering. That's my PhD's in electrical engineering. And I got in this field when I learned about people making gates in living organisms. And I'll talk a little bit more about that in a minute. So just for those of you who don't know, again, just to emphasize, there's this idea that synthetic biology exists as a discipline. And it's supposed to be kind of putting the engineering in genetic engineering. And for example, what we want to do is we want to take pieces of DNA typically that are well understood, put them together in new ways to get novel functions in living organisms. So for example, what I might want to do is design an organism that takes in some sugar and then makes a molecule of interest, but does this with some periodicity, where as high levels of sugar are present, it produces. When that drops down, it waits for sugar to accumulate and then produces the chemical. We might want to engineer therapies that know about what kind of cell types are around, along with other biomarkers, and then produce outputs. Things like circuits in the microbiome that take a certain pH, a location, maybe have cells that die that are potentially hazardous, or produce some controlled process, or of course kind of engineered plants that take into account environmental conditions and produce things like pesticides or drought resistance mechanisms. So the idea is that each of these little symbols are pieces of DNA that are well understood, and we'd like to engineer them. So this idea of synthetic biology emerged. This really plays on this essential dogma that DNA eventually becomes a protein. And I got into this because somebody showed me this little arrow, and they said this was an open reading frame or a gene. I said, that's pretty interesting. And they said, well, the only way for that to work in a bacterial system, for example, is to have a promoter before that's going to recruit RNA polymerase that's going to start transcription. Then that's going to stop at a terminator. And they said, well, in addition, you might have a ribosome binding site, and that's going to help with translation. And so when I saw that, I saw a series of rules. I said, well, in order for this gene to work, we need these parts to flank it in certain orders. And it led me to create a programming language called Eugene, which allows you to take parts in rules and create combinatorial variants of those DNA sequences. And then once I had thousands of sequences, I made algorithms to automatically plan their assembly, and that's kind of spun out to where I am now. So synthetic biology, again, is this idea that we're going to take these parts. We store them in registries now. In fact, there's an entire competition called IGEM, or the International Genetically Engineered Machine Competition, where undergrad students grab these parts and they put interesting tools together. And they were started on this combinatorial kind of chemistry called biobricks, where you could put parts together. And there's a whole industry now based in synthetic biology. So one of the tools that I'm known for is something called Cello. And what it does, again, my background at the end of the day was design automation for semiconductors. And one of the ways that this happens is that there's a programming language called Verilog. Verilog allows you to describe logically what a system could do or should have as a function. It's then synthesized into a set of logic gates. So this is a process where we take Verilog, we parse it, we make it into something called a netlist, and then eventually implements a truth table. So this tool does that. Then in addition, what this tool then does is take this circuit and turn it into an equivalent circuit that's only made of what's called a NOR gate or an inverter. This again has nothing to do with biology, and we do this translation because we have a whole bunch of biological NOR gates. And so the biology comes in, and what this tool does is myself and collaborators at MIT created a huge library of genetic NOR gates. A NOR gate's input was the presence or absence of a small molecule. And if either one of them was present, you would not get an output. That what would happen is a protein is produced which represses a downstream output. And so the problem became taking this circuit, mapping parts on it that gets these high-low outputs as actual genetic circuits, and then we enumerate all those combinations in a tool. So we end up making all of these different variants. So that was pretty interesting. We made all 3 input logic gates. And so people from computing are like, oh, cool, 3 input logic gates aren't very big, so they're relatively small compared to computing, and they function on time scale of hours. So again, they're not competing with silicon, but what they are doing is trying to do those applications I showed earlier. You can't put you can put a chip in your gut, but there are probably going to be problems with that, as opposed to reharnessing the microbes that are already existing there. And so this is an example of one of those logic circuits. And so we made these, and they had, tens of parts and many of these connections. These arrows here, when this gene produces a protein, it shuts down a transcriptional start site, which prevents the production of another protein. And when you have too many of these arcs, they start to interfere with each other. We call this crosstalk. These proteins aren't actually orthogonal, and they have kind of binding sites, other locations that aren't predicted. So what happened for me is I said, well, wouldn't it be interesting if we broke these gates up, and then we put each of these collections of gates into individual cells, and people have done that, where they put the gates in individual cells and had them communicate to one another through intercellular signaling molecules that are used for a phenomenon called quorum sensing, where bacteria, for example, can communicate and say when they come to a certain population size. But the question would then become, how do we know how they communicate? And so I got into microfluidics because I said, wouldn't it be interesting if we could put these cells into individual compartments, connect them, have valves that allow them to communicate. We need to control these valves as a computer. We could actually have them produce outputs that are electronically measurable. We could then have them communicate to one another off chip and control these entire collections of hybrid bioelectronic systems. So the idea was going to be that if I already had a DNA compiler, function in, the DNA out, it would be nice to put the function in and get a microfluidic out. So it's interesting, I'm glad these talks have happened the way they have, because everyone's shown microfluidics, but no one's actually said, how did I design the mask for that microfluidic? Or how did I actually design that microfluidic itself? That's what my group does. We make software to design microfluidics. And so the way this works in electronics is we have this very well understood design flow. What my group does is we make tools to specify, design, build, and test microfluidics. And so we have a whole bunch of tools that I'm going to talk to you guys about that go from high-level specifications all the way down to we start to build and test these. A plug for where we do this, people have asked where we're at. So I'm actually at Boston University. Again, this is a new building called the Center for Integrated Life Science and Engineering, right on Commonwealth Ave, the main strip through Boston University, a major street in Boston in general. Some different views of it. We're actually on this 4th floor right here. And what I have is a lab called the damp lab. It came out, I called it the damp lab because it wasn't wet, it wasn't dry. So the joke was it was damp, but it actually became a pretty good acronym for design, automation, manufacturing, and prototyping. This is the lab manager. One thing we do in that lab is I'm very interested in how we automate the entire kind of molecular biology design process. So if I come out of my design software, Cello, with 1,000 circuits, and I go to my postdoc or ed student, now pipette these, they slowly back away from me and run out of the building. What we have is, and what we're creating here, is a high throughput, very replicable automation system with liquid handling robotics and other labware that executes protocols. But what we also do in that lab is we make low-cost microfluidics. And again, we saw electrowetting, we've heard all these technologies, capillary. We do continuous flow, CNC milled microfluidics using some of the technologies that Will talked about. Again, when we mill out these, we then have controlled valves with pneumatics and syringes. And the point being, because this is coming from a software program, we can curate not only the substrate in CNC milled plastic, but we can also produce the 3D printed labware that surrounds this, that houses the pumps and all of that. So the vision would be that I would have sat down at my computer and said, I want this circuit, and I know that I want it broken up, and it would actually say, well, I need to have a trap for these cells, and I need to put a pump, and then I need to route this input to another input. That would get turned into a microfluidic mask. We'd mill that out, and because of this description, we'd also have the causal relationship between which these valves should open at what point, so we could control them automatically, and then we produce them. So that's kind of the vision. So I'm going to take you quickly through some of these software tools that we have. The first tool, one of them is called Neptune. What it has is we need to describe the microfluidic. So we need to say we have these flows. Flow A goes to flow B. Flow A and B then mix. They go on and create flow C. It's kind of the abstract description of the flows. So it looks like this, where you have a flow, you have a collection of input flows and output flows, and then operations. The operations are user defined. I can say tilde is mix. I can say plus is split, et cetera. So it's sort of human readable. It ends up making a net list. Again, it makes a graph, just like that graph would have come out of CAD tools for electronics. And so we played around with this a little bit. And so this is just an example, a very simple one for something called acoustophoresis, where you vibrate effectively this channel and you separate heavier and smaller particles. It would look something like this. So a video is worth 1,000 words. So this is a video of the software running. So you sit down and you actually type really quickly your microfluidic flow relations. We imagine a flow at some point where you actually don't write this, but there's a higher level tool on top. But right now, you have to say, I have this many inputs, this many outputs. I want the flows to mix in this particular way. It has an additional parameter file where you're specifying what those operators do. The minus is a cell trap. The divide is a valve. You have to define them in some parameters. What's going to happen is when we're done, it's going to translate that description into a lower level description of all the channels, the ports, for the 2-layer device. One layer is flow, one is control. So then you have this intermediate description, which on itself in some ways is an advancement because this is not standardized either. So you have this description along with information about spacing and rules, because depending on your technology, there are different minimum spacing for all of this device. It really is kind of analogous to parts of a VLSI design flow with design rule checking and things of that nature. Then at the end, you end up compiling that. So it's compiling, it's actually placing and routing this device. And so the output of that is shown here. That's the flow layer. So it's saying I have these 3 inputs, I have a valve, then I have some serpentine mixer, and that's the control layer. So it ends up making this microfluidic. And it ends up producing masks that you can then send for soft lithography if you want, or you can actually send directly to the mill for milling out. So that net list is called MINT, for microfluidic net list. So this is another thing that our lab has tried to develop with other folks, is to have a readable, human readable format for what the chip looks like when it's laid out. This lets us exchange chips, it lets us make benchmarks, et cetera. So this description here ends up making this microfluidic. These are mask alignment marks, if you're wondering what they are. So you have flow layer in blue, the control in red, and then there's cell traps. So for different levels. And so the core part of the tool, what it does is once it gets that net list, it has some pieces that are control, these orange boxes, some are flow, the blue, and what it has to do is take that graph and actually move it around to adhere to the design rules that are in the net list and all the different parameter spacing. So right now it's moving them around, respecting those, and also probably in the future respecting some performance requirements once we put a more kind of fluid dynamics aspect to this. So the end result, that abstract graph, gets turned into a place and route net list. So again, that's part of what the tool has to do. So what we've created as well is we have a whole bunch of primitives. We have not only how the pieces connect, but we've created a library of microfluidic primitives you can use and you can stitch together. So these can be those operators. So I can an operator for a rotary mixer or a gradient generator, et cetera. And these are pieces these are primitives that the software can use. So a lot of people don't want to use that LFR. They don't want to write them. They don't want to write textually. They want to use this kind of drag and drop GUI. So we have a tool called 3DUF because we used to 3D print these microfluidics, but we liked the name so much we never changed it. So it's still 3D microfluidics. So here you have a bunch of primitives. This has actually changed quite a bit since this video. So here are your primitives. You're dragging them on and you're connecting them. So right now you're in the flow layer, so you can actually lay out the layer for the flow, and then you can switch and go to control. When we first started doing this, my grad students told me, and postdocs said, what people did is they went to Illustrator, and they would draw these, and I think that's fine. I'm not criticizing that. But the approach would be that you'd want something a little bit more tailored for this. This allows you to do things kind of holistically. So at the end, you're seeing that you can export that design, you can save it, you can select it, you can size everything at once. You can get kind of replicable designs more quickly, I'd like to think. And this is still a work in progress. At the end, it actually exports the SVG file so you can actually fabricate it in different layers. And then the point being, we have these desktop CNC mills. I can actually drag that file right onto this milling software. We don't create that, and then it actually queues up the mill to mill these out. So it's kind of this, we're trying to make this end-to-end flow. So the making part is called Microfluidics. This is again in the upper corner, seeing that we have these desktop mills actually milling out the flow and control layers in plastics. Again, this is this design where we have a layer for flows, a layer for control with the valves that Will described earlier, PDMS sandwiched in between, and we can make these designs. We're trying to standardize this process.
Position and forward throughout.
We don't need the music, but what happens is, so part of this iGEM competition last summer, the students now have gotten this process so standardized that they can do it relatively quickly, and they made a bunch of tutorial videos. So once you get the design files out, there's no robot butler in our lab yet that carries the polycarbonate and puts it on the mill or prepares the PDMS. That's the next step. But we're trying to make it so even that process at least has some kind of a consistent set of protocols. So the students have made this, and we'd like to make the case that people can do this pretty quickly. People always ask me, well, how does this low-cost milling compare to photolithography? Well, yes, it doesn't have the same kind of feature sizes that you might be able to achieve with photolithography, but the costs are much cheaper. And typically when I show someone a microfluidic device, they're like, that's not going to work. And I'm like, good, because we're going to make hundreds of them with different parameters. We're not going to go to you and say, this is the microfluidic chip you must use. Rather, it's an iterative process where we make many different variants and we try different things. And so that's really where I think some of this strength is, that you can come in, make one in one day for less than a couple bucks, and then test it and keep iterating. Yeah?
Where would you put the stereo that took up the 3D printing?
That's a good question. Probably between here. That's my guess. The high-end mills are pretty good in terms of feature sizes, but that's probably where I put 3D printing. So just examples of other things we made. So we have these graphs. I said that the graph data structure of routing fluids around is a key part of our abstraction. Well, if you have a non-planar graph, meaning that I made a graph that I cannot draw without crossing lines, we need a way to cross lines. And so we made a device called a transposer that lets us cross lines. I feel like I should have called it the Ghostbuster device for like crossing the streams. But whenever I tell that to like students, they don't get it because I think they don't know what Ghostbusters is anymore, and the new one didn't have crossing the streams. So maybe it's good that we didn't call it that. But we made a primitive that lets us cross streams. And this was really important because one of the things we want to do is automate this. And so we have a mechanism that when you have to cross them, these are inserted automatically. The same student that made that worked at Draper Labs in Cambridge. And what they did is they said, let's put this whole design flow to the test. They made this device that, again, what it does when it's actually excited is that it separates and it separates particles of different sizes, in this case blood and bacteria. And the issue here was that it's very tough to make a predictive model of how this will work in glass. Well, actually, glass, they can do this. Glass is rigid and it's more expensive. They want to do it in plastic. Plastic is much cheaper, but the model is much harder to make. So instead of spending all this time making this perfect mathematical model, they used our software to explore this multi-dimensional parameter space and create many devices at once and test that. And that's what this paper is about. So another thing we've done kind of with microfluidics is we've designed these genetic constructs that respond to an external molecule called arabinose, and when that's present, they express a gene, beta-galactosidase, which in conjunction with lactose changes the pH of the solution that it's in. So we made this system called Twee coli. This is not in a microfluidic, they're in test tubes. And what we're doing is moving this to a microfluidic where you actually can add these external components. They'll change the pH of the solution. That's detected by an electronic component that then tweets that my pH has changed. It then receives that and then activates a syringe pump here. But imagine this being a much smaller single device. That is, what it's doing is based on the conditions in the chip, it's self-regulating. That's what it's trying to accomplish. The tweeting thing was just cute, but it showed us that we could do it with simple existing electronic communication mechanisms. So we had this idea, we had this setup with an actual test tube, and we showed that these are different pH scales. We showed that we could get a variety of different pHs depending on how we induced the system and it got to work. So it's just a little proof of concept that we're still working on. If you're into synthetic biology, you know one of the interesting things are people are really looking at how to now work in cell-free systems. So imagine you take a cell, you basically break it open, you take the pieces out that are needed for transcription and translation, and then we do this kind of in vitro. This would be interesting to us because this might be more amenable to microfluidics, so we're exploring that in my group as well. And one of the ways we're doing this is with droplet-based microfluidics. So I don't think I need to tell this group much about that, but the idea is that, again, droplet microfluidics could be really interesting. We're already working continuous flow, so we can add new primitives that are for droplets. That'll let us have reductions in volume and better resolution, and then we can do different kind of manipulation at the droplet level. So those videos are not from my group, but these videos are all pictures from my group of primitives that we've created for a variety of things. So the same way you can drag and drop the primitives I showed you on the canvas before, you can now drag and drop droplet generators that do different levels of concentration, different sizes and rates, ones that say basically connect a tube from one microfluidic input to another and let us transport droplets between tubes, mixing droplets where we see the blue and red droplets merging, droplets splitting. We can have droplets be stable off chip, and then we can also have high throughput transport related to this. And what we started to show here are these are droplets moving through our system. What we're able to do is just express protein. So this is just a simple expression showing that we have biocompatible chips. So we've taken that cell-free mix. We've had some control where they weren't exposed to any of the mechanisms that would express protein. The others, we add a particular biological construct and an inducer, and they actually express protein. And we can show that we can get this to happen on chip in environmental conditions and transport these around. So the next part is we're going to be doing this in a more deterministic fashion. So one of the things I showed was we had a CAD platform. You can drag and drop primitives. You can start to specify them at a higher level of abstraction. But then I said we're going to do droplets. And so if we're going to do droplets, we want to say certain sizes and we want to say certain rates. So if I told you that I want a droplet this, some size I give you, how big should the orifice be for oil? Like I have no clue, right? So what we'd like to do is we have another platform that what we did is we made a whole bunch of droplet generators. It's a multi-dimensional space. So there's the size of the input for oil, the size of the orifice coming out, the size of the channel. There are many different parameters we could make. So we made 25 devices that are different in those parameters. And then we specified different flow rate ratios through them and different dimension of capillary number. So we basically made a space of different droplet generators. We stored this data related to the geometry, the conditions, and we record the generation rate, droplet sizes. And we use this to start a machine learning training data set so that all you would have to do is say that I want certain droplets of a size and a certain rate, and it would automatically size your device for you. So it automatically designs the dimensions of that droplet generator to give you the performance that you'd want. So at the end, you just say, I want this rate and this size, and then it will actually place it, size it, and route it together. That's where we're moving. This is not completely finished yet. So the inputs to a tool like this, there are all of these aspects that go into it, but you only specify the generation rate and droplet diameter. And it goes through this multidimensional space and looks at that. And what we're showing is that there were different mechanisms for using that data. These are different mathematical models that determine generation rate and droplet size. We're just showing that some of them have more errors than others. So basically, we've used a combination of these techniques, depending on if you're trying to get generation rate or size. We pick one of those. If you're solving for both or solving for one, we give you ones that end up giving you the ratios you need to actually generate that performance. We're putting that in the tool. So the point of this is we're trying to do some thinking so that when you use a tool, you don't have to do as much thinking. And if you get additional data, there's a mechanism for you to put your performance results in so we can learn from that. And this paper did come out where we took some of these parameters, the velocity of the water, the viscosity of the oil, the density, et cetera. Put these in to something called a neural net to train this kind of learning data set to come out to say how do these relate to the end droplet radius. And we were able to say that we were able to predict the radius based on some of these parameters, and we were able to do this with a high fidelity. And so most of what we're trying to do is make it so that you have requirements you'd like, either the connection of flows, the size of the droplets, or their interactions, and we can ensure those. So someone asked me what our iGEM team is doing this year. So this year what they're doing is we have the microfluidic here that we designed through everything I just shown. So on this microfluidic, cell-free biology is happening with the primitives that we've demonstrated and using the workflow that I've said. At some point, those biological samples we need to put into some larger laboratory environment. Because I'd love to say that I can put this microfluidic in my flow cytometer, but I can't. And I can't put this in a plate reader. I can't put this on any of the other liquid handling robots I have in my lab. So something we need to do is transfer the liquid off of this microfluidic into some standard piece of labware. So what we've done is we've created this active XY plane that very precisely puts the droplets, because we know what rate they're coming off at in particular locations. And we have some microfluidic primitives that let us select of these coming out. We can multiplex the droplets out, and we can move this around. And so this is just a prototype, but what we're trying to show is that I can automatically design a microfluidic. It would be doing what it needs to do. It would have an infrastructure to transfer the results of that into standard labware that we then could use in that large-scale robotic platform I showed many slides ago that my lab also uses to integrate with the rest of our synthetic biology. So we're trying to make this Frankenstein right now that merges microfluidics and traditional kind of synthetic biology. So that's what I do on some level. And then people said, talk about security. I was like, okay. I think hopefully you can imagine many, but I'm thinking one thing is there's a place for secure design. So I said, let me Google what's going on in microfluidics for secure design and look at this paper, lo and behold, from Christian and everyone's group coming out about if you're actually laying out a chip, what kind of security concerns could arise. And so naturally, one thing that could happen, and I think should happen in the design of microfluidics CAD systems, is we should bake some of these rules in. That when I automatically lay things out, I'm taking these rules into account, or if I'm manually doing it, it warns you if you're violating these. So I think CAD software should take that specifically into account. The other piece, this is just a generic idea taken from another paper that was talking about how to make QR codes in microfluidics and biology. But more generally speaking, I think you could use the design software to specifically put in watermarks, specific pieces into there that validate that this is an authentic microfluid. We heard this earlier. I thought that was really nice from IBM, right, where they have these kind of dynamic codes that are generated. That could be baked that could be automatically done with a lot of the CAD software. So I'd love to chat with people about these options. The other thing is just benchmarks. So we should have benchmarks related to security. I'm wrapping up. Right now, myself and Philip Brisk at the University of California, Riverside, just did a quick survey. These are just people's benchmarks. And you see that this is one author of benchmarks. They had so many. This author had so many. They're not all overlapping. The benchmark space for microfluidics is all over the place. I think it'd be interesting to look at benchmarks in terms of this benchmark has so many components. This benchmark has so many components. This has so many connected components. You can characterize the benchmarks as to where they fall, and then you can start to automatically generate different benchmarks. So I think this would be interesting to think about benchmarks related to security. I'm not sure what they would be yet. They would be that this application might be more prone to security attacks than this other one. So we'd want to generate some space of insecure or secure benchmarks and put them through a CAD tool. I think that would be good. We need some way for me to say my tool has passed some security test. These could do that. Biology by design. The other part is someone asks me, how do you know you're not making something that's biologically bad? Like I'm assembling some pathogenic sequence. So what we have, and this is work that I've done in the past, is we have a whole set of rules. I won't bore you with all of the syntax, but these things are like spacer, promoter, ribosome binding sites. These are all the little synthetic biology pieces. You can write rules that look like regular expressions. They can make these graphs that say, if I start at a green node and go to a red, and I look at the parts on the edge and I assemble them in that order, if I do that, I can't break any rule. And you can build these up to quite complicated structures and say that if I started a green and I ended a red and I look at the parts, I'll generate these designs, and these designs will be correct by construction. So what we can do is we can, as a community, encode rules for designs that are safe, or designs that only have particular combinations of parts that we know that have been vetted. We can encode that and have designs that come out of the compiling process that adhere to these rules.